I'm Professor Lori Snyder. This is my inaugural professorial lecture, Overcoming Antibiotic Resistance and Understanding Genome Sequences. My research really splits into two interests: antibiotic resistance, which began during my master's research at the College of William and Mary with Professor Carl Vermeulen. Dr. V and his group discovered that E. coli, when grown at higher temperatures, 40 degrees C, 42 degrees C, which are really fever temperatures for humans, high fevers. E. coli, the LPS-O antigen would reduce, and that made them quite susceptible to serum complement killing. So the reduction in the LPS meant that the E. coli could be killed by normal human serum, and this could be an alternative in cases of antibiotic resistance and kind of harkened back to early investigations before antibiotics into fever therapies. So this got me thinking about what we could do for antibiotic resistant bacteria that went beyond antibiotics and other things that we could do. This continued into my PhD, my interest in antibiotic resistance, and I joined the lab of Bill Schaefer at Emory University, who is a world expert in efflux pumps. So efflux pumps are these multi-protein systems that sit in the membrane of gram-negative bacteria, and they pump out antibiotics. Now, they didn't evolve because humans use antibiotics against bacteria. They evolved because bacteria, particularly the bacteria we were working on, Neisseria gonorrhea, they live on mucosal surfaces, and these mucosal surfaces are bathed in bile salts, fatty acids, antimicrobial peptides, things that are very antimicrobial, so the bacteria will pump them out of the bacterial cell before they can cause too much harm to the bacterial cell. So they're continually pumping these things out. And as you can see in the graph that's on this slide, the wild-type bacteria, FA19, shown with the dark circle, that can survive in the mouse model of vaginal gonorrhea infection quite well, and that's because it has an intact efflux pump. But when we knock out that efflux pump, so it's not functional, as in the two mutants that we created in Bill Schaefer's lab with the triangle and the X down at the bottom of the graph, these don't survive in the mouse model, and that's because the natural mucosal secretions in the mouse vagina are able to kill the bacteria. So there's no antibiotics here that are killing those bacteria, that's just what's naturally made on the mucosal surfaces. At the same time in Bill's lab during my PhD, we were starting to receive genome sequence data on Neisseria gonorrhea. So this was the very early stages of genome sequencing. We were receiving fragments of the genome and looking through it for information on efflux pumps in the genome and also for regulatory elements that might be regulating efflux pump expression in these bacteria. So that got me very interested in genome sequencing and analyzing genome sequences. And that passion carried through into my two postdoctoral research appointments at the University of Oxford and the University of Birmingham, where I continued doing genomics and also bioinformatics. So that's using computers to understand genomes, to analyze them, and learn what's going on within a genome. So then started my research here at Kingston University. So this slide shows a comparison of 3 complete Neisseria gonorrhea genome sequences using the MAUVE Genome Alignment software. And we're able to compare them against one another and see how the different regions have been rearranged between these 3 different strains of Neisseria gonorrhea. And see that there's been quite a bit of rearrangement between these 3 strains as they've evolved independently in different human hosts. So this research was done by my very first MSc by research student, Russell Spencer Smith, who's gone off to do PhD research in the US at the University of Illinois. And this was also in collaboration with a taught MSc student and with Professor Mark Fielder. And we were able to show that there's been rearrangements that are mediated by various repeats in these gonococcal genomes, IS elements, Neisseria-specific repeats, and also by fragments and complete versions of phage proteins. And we can see that there's inversions in the genome, so that large purple region we've labeled 4 there is completely inverted. So in the bottom strain, it's below that medium line there, and that means that it's inverted in that strain relative to the other 2 strains. And there are various other small rearrangements and inversions throughout those strains. In the next bit of research that I'm presenting, this was led by Ruth Griffin, who was a postdoc in my lab for a time and is now an assistant professor at the University of Nottingham. So Ruth was interested in Neisseria meningitidis, which is a related bacteria to Neisseria gonorrhea. Neisseria meningitidis can cause often deadly bacterial infections in contrast to Neisseria gonorrhea, but they're very closely related. So they live on mucosal surfaces and they have a lot of the same genomic features. Now, Ruth was concerned with the vaccine components that had been developed against Neisseria meningitidis serogroup B. So serogroup B had been a problem for quite a long time because we couldn't develop a vaccine against serogroup B because the capsule looks a lot like some components of the human body. So we couldn't develop a vaccine against it. The whole research community knew that wasn't possible. So instead, vaccines had been developed using other antigens within the meningococcus. And one of them is factor H binding protein. So part of Ruth's research actually showed that one of the strains she was working on had a mutation in its factor H binding protein that meant that it wasn't going to become a lipoprotein and wasn't going to be expressed on the surface of those meningococci, which meant that that particular component of the vaccine wasn't going to be effective in that strain. So if the other 3 components also weren't effective, then that could be an avenue for this particular strain or for other strains like it to escape the vaccine. So that's also in collaboration with Professor Andrei Karlashev here at Kingston University and with our collaborator at the University of Surrey, John Joe McFadden. One of the things that I explored in depth during one of my postdocs was phase variation in the Neisseria species. So when we think about regulation in bacteria, we tend to think about things like the lac operon that was described by Jacques Monod, where you have regulative proteins, you have repressors, you have activators that turn genes on and off. But in phase variation, you have simple sequence repeats that undergo slippage events, which means that the genes are on or off by stochastic kind of random events that mean that the gene is on or off based on the number of the simple sequence repeats that are present in the genome sequence. So these occur in Neisseria gonorrhea and Neisseria meningitidis in places like the promoter region. So if they occur between the -10 and the -35, as shown in the top of that figure there, then the polymerase won't bind effectively to the -10, -35 regions, and you get various expression of that gene because it is or isn't transcribed effectively. You can also get the repeat within the gene. So that means that the start codon and the stop codon either do or don't align properly, and you can get a frame shift. So either the full version of the gene is made, or it's a very, very short open reading frame, a short coding sequence, and so the protein isn't made at all. So those were the 2 known types of phase variation when we started this investigation. But then, as we did a more in-depth investigation, we discovered that sometimes these repeats happen later on in the gene. And this investigation included quite a few undergraduate and postgraduate project students, and one of those students was Alan Calder at the time that he was a student, an undergraduate student with us, who is now a PhD student with me, and also Colin Churchward, who was a PhD student with me at the time. And what we found was these later repeats could mediate a C-terminal phase variation, where actually the C-terminus of the protein changes. So a full-length protein is made, unlike in the phase variation that we were used to seeing, where you'd either get the protein made or not made. A protein is made, but the C-terminus changes. And in some cases, this means that you get 2 distinct proteins made, as shown in the final part of the figure there, or a fusion of those two proteins into one. In another PhD student project by Sabrina Roberts, who's now working for the Food Standards Agency, Sabrina looked at chorea repeat enclosed elements. So these are elements that I looked at during my PhD and that Russell actually looked at for mediating rearrangements in the genome sequences, as I mentioned earlier, but Sabrina looked at them a bit differently. And what she looked at was their association with non-coding RNAs in the gonorrhea genome. And she found that these were very closely associated with non-coding RNAs. So what you see with those circles is all of the circles that are lined up there on the left-hand side of that graph are overlapping chorea repeat enclosed elements with non-coding RNAs. So more often than not, quite a lot of correlation between these Neisseria specific repeats, which have a lot of function within the genomes that have been described, and these non-coding RNAs, which seem to be involved in regulating functions of genes within the chromosome as well, that they're associated with one another. So they both seem to be involved in regulating what's happening with genes, promoting the expression of genes, and other functions of what's going on in the chromosome. I had an undergraduate project that I gave to Ferhat Beliglu, and he was supposed to go investigate Cree, and I had a hypothesis. And he came back to me and he said, very sorry, but I didn't find what you'd asked me to find, which was very brave of him for a student to come back and say, no, your hypothesis was completely wrong. But he did say, but I found something a bit weird. I said, well, that's interesting. What did you find? What was weird? And what he found was the Cree elements were inverting, which was really novel because phase variation that involves inversion is known and happens in other species, but had never before been described in any Neisseria. So this was really exciting. And I was really glad that Ferhat stood up and said, I found something weird and your hypothesis was wrong, but we've got a whole new hypothesis to be exploring, because we were then able to describe a brand new type of phase variation in the Neisseria. Now these Cree do have promoters associated with them, so the inversion then can invert these promoter regions within the chromosome. And as you can see in the bottom there, the inversion of the Cree will either drive promotion of the DRG gene in this example from that blue promoter on one end of the Cree, or when it inverts, there's no promoter. So that gene wouldn't be expressed. So interesting stuff with the brand new type of phase variation there. And Sabrina Roberts, a PhD student, again helped with that work. So Sparks funded some research into some antimicrobial work that was headed by Colin Churchward, who was a PhD student in my lab, who I mentioned before. He's now at Imperial. And we were very fortunate that Sparks gave us this money to fund Colin's work because we were able to look at antimicrobial fatty acids. So as I mentioned at the beginning, we knew that efflux pumps could pump out fatty acids. And from my genomic work in investigating what sort of efflux pumps were in Neisseria gonorrhea, I knew that there were some efflux pumps that could pump out certain fatty acids and other efflux pumps that could pump out other fatty acids. And I knew there was probably a space that these efflux pumps weren't going to pump out all of the fatty acids. So Colin tried a large array of fatty acids and their derivatives to see if there were any for which the gonococcus was susceptible. And yes, there were. He came up with 7 that very rapidly, within 2 minutes, were able to kill Neisseria gonorrhea. And not only were they able to kill Neisseria gonorrhea, but then he tested them in artificial tear fluid just to make sure they were going to work on the eyes, because this was our primary concern, that infants born to mothers who have a gonorrhea infection, they will get an infection in the eye, and that can very rapidly cause issues with the eye that can lead to blindness if that isn't treated effectively. So if you have a gonorrhea infection that can't be treated or can't be rapidly treated effectively, then the infant can have loss of vision and can have lifelong blindness. So we wanted to come up with an effective, rapid, safe, and inexpensive treatment for gonococcal infection of the eyes, ophthalmia neonatorum. So we were able to find two fatty acid derivatives that are effective in artificial tear fluid. And then using a couple of different models that we have for eye infection irritation models, we were able to show that these are not irritating to the eyes. So in the circular figures there, those are bovine eyes that we use to show whether something is irritating or not. So these aren't live cows. These are bovine eyes that we collect from an abattoir. So place where they make steaks and hamburger meat, they would take the eyes and just throw them away with anything else that they're not using for human food production. So we would be able to go there and ask them, filling out all the proper paperwork to assure them that we're just going to use them for research purposes, and take these eyes that would otherwise be waste products from human food production and use them for research. So that's really great because these would just otherwise be thrown away, but we can use them in our research to show that these products are non-irritating to the eye. And that's what this has shown, that you can see the strong irritant causes an opacity in the eye. And when you stain it with fluorescein, which is the same thing they use when you get contact lenses to see whether you've scratched your eye with a contact lens. And you can see that the test compounds don't do anything like that. There's no difference from using just regular saline on the eye. And when we do histology to see if there's been any impact on the tissues of the bovine eye, there isn't at all. And the other thing we do is a Hetcam test, where when you crack open an egg, you see all of those membranes that are inside of an egg. Well, that's what we do. We use those membranes and the vessels inside of a fertilized egg to see whether the stuff that's just inside of the lid of your eye would be irritated by, like when you get shampoo in your eye and it really stings and it really burns, whether that would happen when you use eye drops. Because we know sometimes when we use medicinal eye drops, it stings and it burns a little bit. Well, that's allowed. But we wanted to see whether there would be that sort of thing with our eye drops, and we don't see that at all. We don't see any stinging, burning indication from our irritation assays there. So the vessels don't swell up. We don't see any clotting or anything. As we do on our control, you can see that it's gone all blood clots and expanded vessels, whereas in our controls with saline and with our test samples, we don't see any of that. So that was really good news, and even better news, Colin, in collaboration with our collaborators at the University of Southampton, they repeated some of what we did, but with their own strains and their own methods, and shown that their Neisseria gonorrhea strain also was rapidly killed by our monocaprin, our lead candidate for formulation into an eye drop or an eye treatment, and also Neisseria meningitidis. So sometimes, rarely, but sometimes a woman has gonorrhea, but it's actually caused by Neisseria meningitidis, and that can get into the eyes of an infant, and it can cause an infection as well, but this can also rapidly kill Neisseria meningitidis. It can also kill Staph aureus, which can cause eye infections, and Pseudomonas aeruginosa, which can cause eye infections. Now, Pseudomonas aeruginosa, that tends to be from contact lens wearing, so you get a little scratch in your eye from the contact, and it causes an infection there. The Neisseria don't need to have any damage to the eye. They can just cause infection. So they're unique in that way, I think, because they just cause infection when they get in the eye. They can get around all of our defenses on the eye, which is why it's so important to be able to protect the eye health from these infections. Colin and Alan also did a study to see whether resistance could develop to monocaprin. So they grew these bacteria for long term with the monocaprin in the media so sub-MIC concentrations of monocaprin, so not enough to kill the bacteria, but just a bit less than that to see whether they could induce resistance to the monocaprin. And they also did the same thing with myristic acid, which is a different fatty acid, but one which we knew the bacteria had an efflux pump that it would be able to pump it out, to see whether it would induce any mutations or any resistance using something that we knew it should be able to pump out. And what we see, the monocaprin's in blue, the myristic acid is in orange, that the starting culture, and this is showing MIC, so the amount that you have to add to be able to kill it. The starting culture and the non-selective growth, those are just the same amounts. So after growing it just on regular media with nothing in it, nothing changes. Growing it in monocaprin, we see a little bit more is needed to be able to kill. And that's for both monocaprin and myristic acid. And so what we see, and this is when we do the genome sequence and look at what's happened in the genome, we see mutations in things that are a general stress response. So we think that growing on these fatty acids seems to induce some sort of a general stress. And the same is true when you grow it on myristic acid. So in that last pair of columns as well. So we see that same little bit of bump up for monocaprin, and that's the same for its genome there. It's got these mutations in general stress genes. So I think that there's a little bit that these bacteria can go up in their MIC, in their ability to withstand monocaprin, but not to a resistance level, we wouldn't say. What we do see in the myristic acid, when it's grown on myristic acid, we see mutations in FAR-R, which is involved in fatty acid resistance. It's a fatty acid efflux pump, and a big jump up in the MIC for myristic acid. And we see that resistance occurring there and see the expected mutations in the genome there. Colin was able to contribute a book chapter to Neisseria gonorrhea methods in molecular biology with his method for infecting the bovine eye. So you can see that's there. Sorry for anyone who might be squeamish. Again, this is waste product from human food production that we're able to use in our research, and we're able to effectively grow Neisseria gonorrhea on the surface of these eyes, and then they're able to be used for various experiments to show the growth of the gonococcus on an eye, mimicking how it be on a human eye, able to clear it with various potential treatments for gonorrhea eye infections, and that's been contributed out to the research field based on this publication in the book Neisseria gonorrhea methods in molecular biology. And that method was part of a contribution then to our scientific reports paper last year, led by Colin Churchward again, which published our formulation for a thickened eye drop, where we've put monocaprin into the thickened eye drop and showed that it is effective at clearing the eye of gonorrhea growth, and that it is better at doing so than other recommended alternatives to antibiotics for prophylaxis. So one of the other prophylactics ideas that have been proposed is to use povidone iodine. So this is a betadine type solution. And it is cited in the literature that it will cause irritation, and it certainly does in our model, our B-cup model, using those bovine eyes. So what you can see there, we have been able to show complete clearance of gonococcal growth along those top circles there. There's no little dots. Those would be the gonococcal growth after it's been on the bovine eye on 0.25% monocaprin. So that's the concentration in the eye drops that we've applied to the eye. On the bovine eyes on the bottom, if we take 10 times that, so 2.5% monocaprin eye drops, we see no irritation of the bovine eyes. None whatsoever. At 10 times the concentration we're proposing would be needed to completely clear the growth from the eye from just one treatment. using what's been recommended as the clinical doses, so not 10 times, but the normal doses of 1%, 2.5%, or 5% povidone iodine, you see irritation. So normal doses, not 10 times, whereas ours is 10 times, you see irritation as highlighted by those white arrows that are on the slide there. So these are my current PhD students, Afsan Rafiq and Faith Okuchewo, and they have taken the model of irritation and applied it to a totally different alternative antimicrobial therapy. And this is in collaboration with the University of Warwick. Freya Harrison's group has investigated a medieval recipe in collaboration with Christina Lee, who is from the University of Nottingham, and their works on medieval manuscripts and translating these. She has discovered that there are a wealth of different recipes, particularly from something called Bald's Leech Book. And so she has Bald's eye salve, which has been shown to be particularly effective against MRSA, so a very resistant Staph aureus. And we have taken samples of this into our lab, and Afsan and Faith have shown that it is effective against Neisseria gonorrhea, so it is effective against our gram-negative as well as the gram-positive that it's been used against. And we have also applied our irritation models to it. And other collaborators have used it for other things as well to show how it enhances wound healing, etc. And that has all been published in scientific reports last year. But here we show our BCOP model and different batches of the eye salve there. And we've also started using a slug irritation model. So this is a mucosal irritation model. We want to move into looking at whether any of these alternative antimicrobials can be used on mucosal surfaces, if we can develop them for vaginal therapies, etc. And so we've moved into using a slug mucosal irritation model where you look at changes in the weight of the slug over time, and you look at the secretion of mucus by the slug over time. And what you can see there in green is the positive control. So the weight of that slug goes down and down and down as it becomes irritated on contact with your irritant. And in purple, that's PBS, so that's a saline. And so that's a negative control, that the slug doesn't change weight very much as it comes into contact with saline over time. And then the different formulations of the eye salve are shown in the other colors. And you can see that they're not very irritating. They're not quite the same as a negative control, but they're a bit below the negative control there. So we're quite pleased with those results there, and with our collaboration with Dr. Harrison's group looking at these medieval formulations and potential antimicrobials. So on the genomic side, going back to that, we've had a long-term project that was originally funded by the Southwest London Academic Network to look for initially Neisseria meningitidis genome sequences from the throat swabs of student volunteers. Now, we didn't find any meningococci, but we did find 4 Neisseria commensal species, and we've genome sequenced them and done some really in-depth analyses of these strains. And one of the things that Alan Calder and that Jude Menchidi has found is this region of these 4 different commensal species, where in 3 of them there's a capsule locus. So usually capsules we think of being in Neisseria meningitidis, but here we have capsules in non-pathogenic Neisseria species. And it's becoming more apparent that some of the non-pathogens have got capsules as well, and that they are therefore a reservoir for capsules to horizontally exchange between the non-pathogens and Neisseria meningitidis. So this might be a place where there is some horizontal gene exchange between the commensals and the pathogens. And there are also various other new islands that we're exploring that both Alan and Jude are exploring in different avenues for these strains, as well as the various other projects that they're investigating. And they're both current PhD students in my group. So there have been a lot of people involved in these projects. These are just the published projects. There's been a lot of undergraduate and postgraduate students, as well as visitors in my lab over the years. But these are the names on the left and in the middle of Kingston University research students, undergraduate students, master's students, and also staff who have been involved in work that has contributed to the research that's gone on in my lab and has contributed to the various research projects that I've mentioned here, and the various funding sources that have contributed either to funding the research or to enabling the research to be presented at conferences. And on the right-hand side are some of the collaborators that have been involved in the publications that I have described here.